All right, here we go.
Welcome everyone who just joined us. We are going to begin in just a moment. We're just waiting for people to file into our virtual meeting space. All right, I think we will get started. I am going to give you a few pointers. I'm Juliana Medeiros. I am a scientist here at the Holden Arboretum, and I am going to be one of your co-hosts tonight, along with Miss Maya Allen, who you are going to meet in just a moment. I just want to give you a few technical notes about the meeting. First, just so that you're aware, the meeting is being recorded and it is also being live streamed to YouTube, where it will be available for public viewing. Sorry about that. I got interrupted by my computer. The audience microphones are going to be muted for the entire talk, and we also recommend that you turn off your camera just to save bandwidth. We do encourage you to ask questions, and for that we're going to be using the chat feature. Ms. Allen will serve as our moderator tonight, and she will be monitoring the chat window for your questions. So if you have a question, put it in the chat and we will ask our speaker after the talk. So we are getting close to starting our talk, and before we do that, I would just like to welcome you again to Holden Forests and Gardens. And our mission here at Holden Forests and Gardens is to connect people with the wonder, beauty, and value of trees and plants, to inspire action for healthy communities. And we are really, really proud today to be partnering with Black Botanist Week to bring you this, I believe this is the 9th installment of this very exciting year-long series entitled Growing Black Roots, the Black Botanical Legacy. This series is covering a broad range of botanical disciplines and delving into the historical legacy of formally trained and self-taught Black botanists. Through this series, we seek to shine a light on the Black roots within botany and to foster a community of Black botanists, and to demonstrate the diversity within this community and inspire others who may not have considered botany as a career choice. We want to give a gigantic thank you to the Cleveland Foundation for sponsoring this lecture series. The Cleveland Foundation works to enhance the lives of all residents of Greater Cleveland, now and for generations to come, by working together with donors to build community endowment, address needs through grant making, and provide leadership on key community issues. You can learn more at theclevelandfoundation.org. Now, without further delay, I am going to introduce my co-host, Ms. Maya Allen. Ms. Allen is one of the founding members of the Black Botanist Week and also a graduate student at the University of New Mexico, where she is studying the ecology and evolutionary biology of the role of phenotypic plasticity in colonization success. Ms. Allen is going to tell you a little bit more about Black Botanist Week and introduce tonight's speaker.
Thank you, Dr. Medeiros, for that introduction. We are very excited about this partnership with Holden. Black Botanist Week was founded this past summer to promote, encourage, create a safe space for, and find more Black people and BIPOC who love plants. Our speaker tonight is Jade Bleau. Ms. Bleu's research focuses on understanding how plants respond to environmental stress, particularly how they respond to pathogen infection. She has a diverse background in different aspects of botany, including investigating palm tree morphology at Kew Gardens, examining the molecular biology of pathogens that attack vegetables, and most recently, elucidating the role of oxidants and antioxidants as signaling systems in response to environmental stresses. She takes a special interest in research that has direct application to real world problems, working with nonprofit organizations such as CABI, which stands for the Center for Agriculture and Bioscience International, to affect data-driven decision making. Her doctoral research as part of the East Bio program, which is the East of Scotland Bioscience Doctoral Training Partnership, in the SPOL Research Group focuses on the mechanisms of redox signaling in the plant immune response. So without further ado, take it away, Ms. Bleau.
Thank you very much, Maya, and thank you very much, Maya and Juliana, for organising the Black Botanist Holden series. So hello everybody, thank you very much for coming to the talk this evening. I'm Jade and I'm currently a final year PhD student at the University of Edinburgh, and tonight I'm going to talk about reactive molecules that we find in plant cells, the deadly messengers, and how plants have evolved to take advantage and utilise these as signalling tools. But before I go straight into the details of what I work on, I thought I'd give a bit of an introduction to kind of my journey into plant science, or like my botanical journey so far. So I was born and raised in London, and I originally actually wasn't that interested in plants, which I feel like is almost blasphemous to say, but I was really interested and obsessed with David Attenborough documentaries. And my original goal in life was to be the next David Attenborough and save all the animals. But then I went to the University of Leeds to study biology, and they have an excellent plant science department. So this started to pique my interest in some of the courses I was taking. So while I was there, I did an internship over summer at Kew Gardens when I was home in London. And I was very lucky, and I got to work in the Palm House for all summer, and I looked at different palm. Can you hear me okay? It said I was muted.
You went out after you said, I worked with different palm.
Oh, okay.
And now you're back and we hear you.
That's fine then. Yeah, so then I worked with different palm trees from different habitat types, and worked on different morphologies to see how they differ depending on where they would naturally grow. And this kind of got me thinking about how plants interact and adapt to their environments. So I went back to Leeds, and I was there for my final year, and I did a really interesting course called Sustainable Food Production, and that largely focused on the challenges we're facing at the moment, and that kind of basically put me on the path that I am at the moment. So just like a quick overview, just like, I don't know if it's like something you've heard before, but this is something that I kind of heard about, and then I found quite interesting, and this is what I've kind of has led me to where I am today. But basically, one of the biggest challenges we're facing is that we need to improve the amount of food that we are producing to feed a growing population. And over 2,000 of our daily calories come from like plants and plant-based foods. And this is forecast to grow even further. Hold on, I'll put my laser pointer on. And this is forecast to increase as the years go on. But one of the limitations that we have is that we can't really increase the amount of land that we use to do this. So already of the habitable land on Earth, 50% is used for agriculture. And even though plant-based foods supply over 80% of our global calorie supply and over 60% of our global protein supply, we only use 23% of the agricultural land available for crops, and the rest goes on livestock and meat and dairy. So one of the major questions is that how do we feed a growing population without using more land, but also how do we do this in a sustainable way? So essentially by lowering our emissions and our carbon footprint. So obviously we can, one of the ways we can do this is improve the foods and the feeds we give to livestock. We can, if and where possible, go to plant-based diets or move more to a plant-based diet. But the other option, which I'm particularly interested in, is making resilient crop breeds. So this then led me to go to start my master's. So I started at the University of Sheffield, and I was very lucky to be able to go to the land of tulips to do my research project. So I went to the Netherlands. And while I was there, I worked for a vegetable breeding company called Enza. And while I was there, it was great, and it was a very good place to work. And I worked on the priming of crops and plants against pest and pathogen attack, which was very interesting. And I really enjoyed it because it was very applied side of science, and I got to see kind of work that was done in the lab in a more like real world setting. But because I was only there for 8 months, I didn't get to fully dive in. And at this point, I wanted to learn and study more about how exactly plants combat environmental pressures and what exactly is going on inside the plant. So then this led me to find my supervisor now, Stephen Spall, at the University of Edinburgh. So much to my parents' displeasure, I moved even further north and started my PhD. So currently I'm working on how plants respond to environmental stresses. So plants are subject to a range of environmental stresses, and these include abiotic stresses such as flooding, light stress like UV stress, heat stress, nutrient availability, and drought. And then there are also biotic stresses, so the living stresses such as pathogen infections, such as like bacteria, fungi, but also insect pests. So the question I'm asking is, how do they cope with environmental stress? And in the lab, how we study this is we use, I don't know how many of you have heard of this, so I just thought I'd include it just in case there's anybody who hasn't, but we use the model organism Arabidopsis thaliana. And this is very much a model organism for molecular plant biology. It's a very good tool to study plants. It's a member of the Brassica family, which is related to a lot of like leafy vegetables that we would eat, such as cabbage. But it's got a smaller genome, and which was sequenced in 2000, and it's very well annotated. So lots of the genes are described, so we can understand a lot of what's going on. It's very easy to grow in the lab. As you can see, it just tends to grow anywhere in the cracks of pavements. You've probably seen it. And it has a relatively quick life cycle. So if we're making transgenic or genetically modifying any plants, then in theory, it should be quite fast. So on to the main topic. So the deadly messengers, also known as reactive oxygen and nitrogen species, or ROS and RNS. These accumulate in response to several biological processes, and they're very, very common throughout the cell. So they're involved in just like growth and development, abiotic and biotic stresses that I described before. Photosynthesis, there is a big producer of ROS and RNS, and also hormone signaling within the plant, so like long range signaling. But what exactly are reactive oxygen and nitrogen species, and where are they found? So they are free radicals that are formed of oxygen and nitrogen, as their names suggest. So these include hydrogen peroxide, superoxide, and nitric oxide, and these are just a few of them that I've just included now. And they are produced throughout the cell. So they're produced in the apoplast, where you can see that, so they're produced outside of the cell, and then these can send signals inside of the cell. They're produced in the cell wall as well, which can change kind of the structure of the cell wall. So for example, if there's a pathogen attack, the plant could change the structure of the cell wall to maybe stop any more coming in. The chloroplast is a big producer, as I said, for photosynthesis, and mitochondria for respiratory reasons. the peroxisomes, which produce a lot of ROS and RNS in response to metabolic processes, but also the cytoplasm, the ER. So basically, it's very common throughout the cell. And the levels of reactive oxygen and nitrogen species are tightly controlled to the levels are tightly controlled, and to a large extent they're byproducts of metabolic processes. And it was initially thought that they were only negative products of metabolism, but we now know that if they are regulated, they can be used as signaling messages in the cell. So this can be between cells, so intercellular, and this could be, for example, if a pathogen or some kind of stress is just on one leaf of the plant, it could send a message to a leaf somewhere else that isn't aware of this problem yet. So it will just get them ready for it and be like, like you're next almost. So as I said, the levels are tightly controlled and they're usually at quite low levels in the cell. But problems can start to occur when accumulation of reactive oxygen and nitrogen species get out of control. So stress-induced ROS and RNS can have detrimental effects to the plant cell. So for example, in response to a pathogen infection, you can see here that with the green dots, you get an accumulation of nitric oxide upon pathogen infection. This is just a staining method you can use to detect reactive oxygen species. In this case, it's hydrogen peroxide. And yeah, you can see this dark brown area here is the accumulation of reactive oxygen species, but you can also, in severe cases, this can lead to like the complete cell death and collapse of the cell, which obviously, if this were to happen in a crop, would not be ideal. So plants have extensive antioxidant systems, which I will talk about more later, and these can directly remove or essentially detoxify reactive oxygen and nitrogen species, and this can act as a buffer and limit these detrimental effects. However, on a more molecular level, they can cause what we call oxidative post-translational modifications, or PTMs. So post-translational modifications of proteins they're modifications of proteins after they've been translated. And there are many different kinds of PTMs, and oxidation is just one of them, or necessary to go into today. But basically, having a post-translational modification adds a layer of regulation and control of processes going on in the cell, rather than just moderating the protein levels or the gene levels or the levels of expression in plants or plant cells. You can have the protein there ready to go and just alter what it's doing. But oxidation specifically can alter the conformation, so the shape of the protein, the function of the protein, its activity, and where it can be found in the cell. And oxidation specifically usually occurs on the thiol residue, so the SH of the amino acid cysteine. And we call these proteins that are more sensitive to oxidation redox sensors for reduced and oxidized. So if there's a rapid accumulation of unwanted reactive oxygen and nitrogen species, this can cause undesirable effects or undesirable changes, because if you have a rapid accumulation and an important protein to some kind of, metabolic process that's going on is oxidizing, then suddenly becomes inactive, then that can be very detrimental to the plant. But what's quite cool about this, or what I find very interesting about this anyway, is that these modifications are largely reversible. So essentially, what you now have is an addition, like another layer of regulation, and the oxidation and reduction of proteins can essentially be used as a molecular on-off switch if the reversal is controlled. So the reversal of these modifications, and this is basically what the rest of the talk is about, is so the reversal of these modifications can largely be attributed to a superfamily of enzymes known as thyrodoxins. So this is just like a simple schematic of what thyrodoxins can look like. So they're a superfamily of what we call oxidoreductases. So they reduce the oxidation of these proteins, and they are characterized by this active site, so where the reaction between the target substrate and this enzyme happens. Yeah, so they're characterized by this active site sequence, and specifically these two cysteine residues here. And you have a more classical thyrodoxin, which is small and only has one active site. And then you have larger thyrodoxins like these nucleoredoxins here, which have multiple active sites. And there are, as I said, there are many different subfamilies in here, but we won't go into all of them today. So thyrodoxins are very interesting, particularly plant thyrodoxins, because there are so many of them. So humans and yeast have about maybe like 1 or 2, whereas Arabidopsis has at least over 20 that have been characterized in even more thyrodoxin-like enzymes. So as you can see here, they are localized throughout the cell, and you have in some places you have a large number of thyrodoxins in the same place. For example, the thyrodoxin H family and nucleoredoxins are largely found in the cytosol. And then you have the F, M, and Y families, they're all in the chloroplast. So a common narrative is that they are all redundant for each other. And if one, for example, if one is not working, then the other one will just take over. But in the lab, we question that, although this might be the case to a certain extent, wouldn't it be unnecessary for the plant to have so many proteins in the same place with the same function? So it's perhaps having so many in this one place suggests that there is a level of specificity in their responses. So the question that we ask, and I largely asked during my PhD, is, do thyrodoxins selectively regulate stress responses in plants? So the idea behind this is that you might have different stresses. For example, you'd have a pathogen infection, or you might have drought or flooding, and these would induce the accumulation of reactive oxygen and nitrogen species. So these could be different kinds of ROS and RNS, or it could be the same kind, we're not sure. And these will oxidize target proteins, and these are probably different target proteins depending on what the stress is. And what I guess our hypothesis is, and what we have kind of shown before in the lab, is that thyrodoxins could selectively reduce these proteins, specific proteins, to get them active again and get them functioning in different stress responses. So it could be that a thyrodoxin is in a specific pathway that's only involved in ROS signaling. It could be that there are some that are involved in ROS and RNS signaling, or it could be that there are only ones involved in RNS signaling, again, specific to different stresses. So the example that I'm going to give today is the nucleoredoxin family, because this is the one that I've worked on most throughout my PhD, and I think it's quite a nice story. So nucleoredoxin 1, this is one of the 2 NRXs in Arabidopsis. So as I showed on the previous slide, the wild type protein just has this regular 2-cysteine in the active site. But what we can do in the lab is we can generate what we call a recombinant protein. So we can synthesize this protein in bacteria, and you can change it so that the second active site, you change the amino acid of the second active site. And why this is quite useful is because thyrodoxins tend to work on what we call a dithiol mechanism. So you have your your reduced thyrodoxin and your oxidized protein. So in this case, it's a disulfide bond, which is one of the more common oxidative modifications. And then what happens is the first active site, cysteine, will reduce one of the cysteines on your protein. And then this forms this, what we call a disulfide intermediate. And then the second active site, cysteine, of the thyrodoxin will then reduce the second one. So then you get an oxidized thyrodoxin, but a reduced protein. And then your protein can go on and do whatever it was doing before. But what we were interested in knowing is what are the targets of NRX1 in response to stress. And this was done just before I started in the lab. So the method that was used is that you created this mutated protein with the change in the active site sequences, and you can just run your plant protein extracts over it. And because it's mutated, it's stuck in this disulfide form. So any targets of this protein get stuck here. And then we used an approach called mass spectrometry, where you can identify the proteins that are bound to the thyrodoxin, so the nucleoredoxin. And one thing that was really interesting is that it was found that a lot of the proteins that were pulled down were involved in the hydrogen peroxide detoxification pathway. So the details of the pathway aren't super important, because this is just the kind of the catalase section that you need. And catalase is an antioxidant which is highly active in plant cells. So, this should say CAT3, not CAT2. But yeah, so CAT2 and CAT3 are highly active in the leaves, CAT2 specifically, and all three of these were detected as targets of NRX1. So what happens is hydrogen peroxide just detoxifies hydrogen peroxide to water and oxygen. And this was confirmed by looking at if you treat plants with methylviologen, which is the oxidative stressor that I'm going to mention a bit later, and also a pathogen, which is abbreviated to PSM here, but it's actually Pseudomonas. So it's a bacterial pathogen that's a biotroph, so it feeds off living cells. And what was found is that if you knock out this protein, so NRX1, you can decrease, what is found is that the catalase activity is decreased. So this is in response to just a chemical that induces an oxidative burst, and then this is in response to a pathogen infection, and you can see it's also reduced here. And then this is just the control of what that we use for the catalase, the catalase mutant, so that obviously there should be none or very little catalase activity. And then what was also found is that if you use this kind of recombinant synthesizing bacteria protein, you can rescue the catalase levels back in the NRX1 mutant where the NRX1 gene is knocked out, if that makes sense. So if you incubate it with the wild type protein, you see an increase in catalase activity compared to if you incubate it with the mutant protein. So this is the one with the mutated cysteine residues, and you can see here that this is a lot lower. And you can see again even in like wild type plants, adding extra NRX1 boosts the catalase activity. So it boosts the antioxidant activity in the plants, which is quite cool. And again, this is introduced, this is treated with the oxidative stressor, methylviologen, and the pathogen infection as well. But when I started in the lab, so this was done by the time I started, and the question that I then wanted to look at was, in the absence of these antioxidants, does NRX1 maintain antioxidant activity? Can it rescue this, what we call oxidative stress, when the cell becomes highly oxidized because of the rapid accumulation of reactive oxygen and nitrogen species? So I look at this catalase mutant. So because this was detected as a target of NRX1, we thought, what would happen if you got rid of it? So this is catalase deficient. so sufficient in this enzyme. And then I also look at a separate mutant in it's called PAD2. So this is deficient in glutathione, and this is a separate antioxidant which can scavenge reactive oxygen species and reactive nitrogen species. And it's very important because it is known for maintaining what we call the redox status of the cell. So it determines whether the whole cell is in a reduced or oxidized state. And by removing these antioxidants, you get a hyperaccumulation of reactive oxygen species or reactive nitrogen species as well. And so then it means that you can see a response more clearly. So I wanted to answer if thyrodoxins are selective in rescuing oxidative stress in these two mutants. So I wanted to see if thyrodoxins would rescue stress in one mutant but not the other, or if there are ones that have the overlap. And how we do this is, I've done it for a few thyrodoxins, but I'm only going to explain this one today. But we overexpress thyrodoxins in these plants. So you kind of give them a booster so you can see the phenotype, so the behavior of the plant more clearly. And one of the ways we can do this is that we can induce a stress response that causes ROS accumulation. So as I've spoken before about this chemical methylviolin, it's also known as a herbicide, paraquat. So I don't know if anyone would have heard of it that way. And it essentially causes a oxidative burst, so accumulation of reactive oxygen species in the chloroplast and the mitochondria. And in wild-type plants, this doesn't seem to have much of an effect. But if you put them in these redox mutants, so it doesn't have the machinery to get rid of the reactive oxygen species, this disappears, and so it can't deal with it, and the leaf just collapses. And so we thought that by overexpressing thyrodoxins, in some cases it might rescue this, and in some cases it might not. So in this case, this is a thyrodoxin overexpressing line that doesn't rescue it, but then we also have some that do. So you can see here again, the cell death response is very minimal after treating with this oxidative stressor. And in the redox mutant, the leaf again is completely collapsed. And then you can see in this thyrodoxin that there is a level of cell death, but it's nowhere near as bad as the redox mutant. So these differences suggest that there is a selectivity and a specificity of thyrodoxins in rescuing stress responses. And what we can do with this is we can quantify this in the lab. So if you treat with your oxidative stressor, so the methylviolin, MV, and you can make little leaf discs and float them on water. And over time, the cells die. So they will leak ions or electrolytes into the water. And we can measure this over time, and that will basically tell us the rate of cell death that is occurring. So in the catalase mutant, you can see that the black line is just the wild type cells dying over time, and this is a lot higher in the catalase mutant, and similarly the same colours, so wild types dying over time, and then the PAD2 mutant, so this glutathione deficient mutant, is a lot higher than wild type. But what was really cool to see is that if you put this thyrodoxin, so the nucleoredoxin 1, into these mutants, in the catalase mutant, this kind of brings it back down, or like to similar levels as wild type, and then, but in this glutathione deficient mutant, it's similar or slightly higher. So then we thought this was quite cool. So, but how is this, I guess, more biologically relevant or relevant in real life terms? So what's interesting is this chemical that we use, methylviolin, the kind of burst it causes is very similar to light stress. So what we found, which was really interesting, so we put plants in a very minimally higher light intensity than what we would normally grow plants in. And you can see here, compared to wild type, because the catalase mutant doesn't have the antioxidant machinery of catalase anymore, you get these cell death lesions that appear, and this is the plant essentially dying, but you don't see this in your wild type plant. And then when you put this into when you put nucleoreductin 1 into this background, you see there is still a few cell death lesions, but it's a lot less than the catalase mutant. And we can test this by qPCR, so measuring the level of gene expression of what we call oxidative stress markers. So these are genes that typically are always upregulated or downregulated in response to oxidative stress, or this ROS burst. And you can see here that compared to wild type in the catalase mutant, both of these are increased to different extents, but they're both increased. And then when you put this thyrodoxin in, you can see they're brought back down, which is quite cool. So then we wanted to see what exactly is happening. So these oxidative stress markers, there's quite general markers. So we wanted to see what specifically NRX1 could be regulating. So we did an RNA-seq experiment. So we looked at the changes in gene expression across the whole of the Arabidopsis transcriptome, essentially. And we could see that we didn't get any like full rescues, like rescues that were brought all the way back down to wild type levels. But we did see what we call a partial rescue. So you can see here, this is just an expression profile that shows the intensity values of the reads that we got. And you can see here that there are a bunch of genes that are upregulated in CAT2 compared to wild type, or downregulated. And then these are brought partially back to wild type levels, but not the whole way. But this seems to be enough to partially recover this phenotype, which was quite interesting. And then this is just a heat map to kind of illustrate what this graph shows. And you can see that compared to wild type, there's a lot of changes in the catalase mutant. So these are genes that are upregulated compared to wild type, and these are downregulated compared to wild type. And then apart from this cluster, which is just wildly overexpressed, you have a lot of genes that are just brought back to the middle, and this seems to be enough. So then we thought, we looked further into what these genes are, and these are just like a few kind of general categories that we found that are quite interesting. So we found redoxins, which are just essentially, we found other thyrodoxins. We found some that are involved in intercellular signaling. So this essentially systemic signaling that I spoke about before, where you can tell different parts of the plant that might not be in trouble, that they might be in trouble soon, so that they can kind of prepare themselves. Chloroplast transport, which is not that surprising because it's to do with light stress and photosynthesis, would be related to that. And then we also found some membrane receptors, so it tells you kind of from the outside of the cell, it's perceiving the stress, and then it will tell the membrane. So yeah, so it's kind of a more of a short talk, I guess, than the others perhaps, but to conclude, plants have extensive antioxidant systems, as this shows. The details of this don't really matter, but it's just to show that there's a lot of antioxidant activity going on in different plants of the cell. And plants have utilized the oxidant and antioxidant systems as signaling tools. So they've taken something that should really be very deadly to them, and they've kind of taken advantage of it and harnessed it for themselves, which is really cool. And thyrodoxins specifically provide reversibility of oxidative modifications. It provides protection of antioxidant enzymes, and we've shown that it provides selectivity and specificity of rescuing stress responses. So with that, I guess I'd just like to thank my lab, specifically Stephen, my supervisor, and this was a fun photo back before we all had to start socially distancing. And I should also say this is also with our shared lab group that we do lots of fun stuff with. So yeah, so thank you for listening, and I can take any questions. I should also say that because it's almost 1 o'clock in the morning, I probably did go quite fast and missed some stuff, so I'm happy to answer any questions that you have.
Thank you, Ms. Bleau. That was a wonderful talk, and I feel like I actually understand the whole process way better than I ever expected. So that was really excellent.
Fine.
We have some very good questions for you in the chat. So I'm going to turn it over to Miss Allen to get some of these questions answered.
All right, without further ado, your first question is, this is from Sarah. She says, it seems from the metatranscriptome that nucleoredoxin expression induced expression in other thioredoxins.
Yeah.
Does this suggest a level of redundancy in thioredoxins that you mentioned earlier? Or do you think there is specificity of these enzymes?
So I think it could be both. So as much as I like to kind of promote the specificity of thyrodoxins, we do have to admit that there is a level of redundancy because they are very similar proteins, so they, in theory, they could function to act redundantly for each other. But one of what could be quite likely is that you could have, so at the end of the kind of mechanism of thyrodoxins that I showed, the thyrodoxin then ends up reduced, and this has oxidized, sorry, and this has to be reduced in some way. So this could be that so we know it's reduced usually by like an NADPH system, but it could also be reduced by other thyrodoxins. So then in theory, it could be a specific target for that thyrodoxin, and it could just end up as some kind of chain reaction of thyrodoxins.
All the possibilities. That's promising for future research, right?
Yeah, definitely.
So your second question is, do other model plant systems like maize, rice, or moss have high similar copies of TRXs, or it's due to duplication specific to core eudicots or brassicaceae or other specific clades?
No, so the thyrodoxins are in all, I think, all plant species. So yeah, all your like cereal crops also have them. They've been shown to be involved in lots of stress responses as well, like pathogen infection, drought, flooding. So yeah, it's not just the brassicaceae type families.
Wonderful. We have a similar question to that. Is that do plants have similar reactive oxygen species tolerance, or are there more tolerant clades or particular species?
Oh, that's a very good question. I actually don't know the answer to that one, but I imagine different varieties do have different tolerances. That would just be kind of a educated guess, but I'm not sure. But that is actually a very good question. Something I should look into.
And Norm asks, are all the geraniums I have ever grown lose leaves on a regular basis as the plants grow a stress response, or is this a natural type of growth in geraniums?
I don't really know specifically about geraniums, but it could just, if it's a common response you're seeing, then it could be just something that happens as plants get older. They go through like a cycle where the leaves senesce, so they die and then they regrow, so that you don't, older leaves aren't taking up energy that younger leaves need. So yeah.
And also, in addition to your light treatment, is the gene also responsive to other stress signals?
Yes. So it's responsive. So the kind of introductory slides of the nucleoredoxin I showed is in response to pathogen infection as well. Yes. But in the catalase background and the PAD2 background, actually, it doesn't seem to be involved in the immune response if you get rid of those. So it could be that its response in the kind of wild type plants is specifically dependent on the catalase system.
Okay. People talk about antioxidants in plant foods, like vitamin C being very good for you. Are the antioxidants that we eat also used by the plant to deal with oxidants?
Yes, they are. They are indeed. But yeah, they're very good. You should eat lots of foods with antioxidants in them. So yeah, the antioxidants that we are told are good for us are also good for the plants.
Oh, that's awesome. Just another commonality. All of life on Earth.
Yeah.
So you have a compliment. Great talk. And the question is, do you think some specific secondary metabolites also help the plants to cope with environmental stress?
Oh, definitely. I think so. Yeah, I think it's in general, plants just have many different pathways and signaling systems that I don't think there are that many general responses. I think things do tend to have specific roles, especially when there are so many similar ones. It doesn't seem to make sense to all do the same thing.
So to our audience, we have a few more questions. If you are here in the Zoom, please feel free to put your questions in the chat. If you are on YouTube, put them into the chat box on YouTube. And your next question, Jade, is you mentioned that Arabidopsis is related to food crops like cabbage.
Mm-hmm.
Will these experiments be repeated in cabbage once you find an interesting signaling system in Arabidopsis?
Not by me, because my PhD ends in less than a year, but it is a possibility that you could put these similar systems into crops to see if you get the same response.
That could be an interesting future for any students on the call.
Exactly.
And then speaking of which, do you mind if we share your Twitter handle in the chat so people can contact you?
Yeah, that's fine. I'm happy to answer any questions by Twitter.
Okay, wonderful. We will do that. And then your last question of the night, I am not seeing any more questions, is have you exposed different life stages to your light or different stress treatments?
I haven't, but just from, I guess, observations throughout my PhD, it tends to be that older leaves tend to be more susceptible to oxidative stress in response to different stresses that we impose on them.
Oh, interesting. Well, thank you so much for your time.
Yes, thanks for having me. Thanks everybody for coming.
Great questions. And so older leaves and older people are more susceptible to reactive oxygen stress. Yeah. It's funny to think we are so similar to plants. So that's very cool. That's all we have today. I want everybody to give a virtual round of applause for Miss Bleau. That was a great talk. This video is archived on the Holden Forest and Gardens YouTube channel, so you can return to watch it anytime, and please share it with your friends and your colleagues. We have educational materials developed to go along with this lecture. There is a role-playing game on reactive oxygen species, so if you wanted to get into biology theater, now's your chance. And you should definitely check out these materials. And also, before you go, I want to remind you to sign up for the next talk on July 14th. We are hosting Mr. Rupert Koopman. He is the conservation manager at the Botanical Society of South Africa and the chair of the Fynbos Forum Committee. He's giving us a talk entitled From Fynbos to Savannah and Everything in Between, Plant Conservation in South Africa. So you don't want to miss this, and a big round of applause for Miss Bleau. Thank you, everyone, for attending. Thanks, and get some sleep, somebody said.
I'm going straight to bed.
Awesome. All right, well, thank you. It was really fun. It was great content. So we really appreciate you staying up late with us.
Oh, thanks for having me.
All right. We'll see you guys next time.